So if the concentration of sodium hydrogen carbonate is more, then it will provide more carbon dioxide. If the concentration of sodium hydrogen carbonate is less or low, then it will provide less carbon dioxide to the plant. So this is why they have taken 5 separate samples, 5 pond plants they have taken, and with those 5 separate samples or plants, they have used separate concentrations of sodium hydrogen carbonate, so that in all those 5 setups, all those 5 apparatus that they have made, they will provide separate concentrations of carbon dioxide to the plant, which will, so that you can compare the rate of photosynthesis in all those 5 samples. So this is the main point. Can you understand this one?
Understood. And can you tell why they have used that stopwatch?
I'm telling you. First, you tell me that up to this, did you understand?
Yes, ma'am, understood. To see the concentration that carbon
5
dioxide.
different setups. This is one setup that you can see in this picture. Like this, they have made 5 setups with 5 pond plants, and in those 5 setups, they have used the same solution, sodium hydrogen carbonate solution, but the concentration of sodium hydrogen carbonate is different. Why? Because if the concentration of sodium hydrogen carbonate is more, it will provide more carbon dioxide. Then the rate of photosynthesis will be different. If where the concentration of sodium hydrogen carbonate is less, that solution will provide less carbon dioxide to the plant, and it will
be less.
show another rate of photosynthesis. So to compare the rates of photosynthesis, they have used the different concentrations of sodium hydrogen carbonate. Now you see the student counted the number of bubbles. So why they have counted the number of bubbles?
And to see the rate of photosynthesis.
Mainly while photosynthesis
What does this bubble indicate? What does this bubble indicate?
Photosynthesis.
What do the bubbles
Oxygen is coming out.
Oxygen is coming out. Yes.
Yes. So bubbles will indicate that the oxygen is coming out. So bubbles, because the oxygen is coming out, the gas is coming out, and within that solution, it will always come out in the form of bubble. So the more oxygen will be produced, the more bubbles will come out. That will indicate the rate of photosynthesis. Okay. So the more bubbles will be there, the rate of photosynthesis will be. The less bubbles will be there, the less rate of photosynthesis will be. Now let's see what is the next part of this question.
Mainly the question, the bubble all time will represent oxygen. It can be the other gas.
No, other gases, what will come out? What gas will come out after photosynthesis? You tell me.
Yes.
Carbon dioxide plus hydrogen, it makes glucose, that is the food for the plant, and oxygen. So oxygen is the main gas that is being produced. So when oxygen will come out as gases are lighter, so they will always tend to come to the upper layer, right, of the solution. So when they will come out in the form of bubbles. So the more bubbles, the more the rate of photosynthesis. The less bubbles, the less the rate of photosynthesis. Now they have done one thing, that the students counted the number of bubbles produced by the pond water over a period of 5 minutes. So just to count that time, to get the record over a particular time period, they have used the stopwatch. If they would not use the stopwatch, they could not count that time period, right? So suppose they have used 5 different samples or 5 different setups, and they are counting the number of bubbles. So they must have a uniform time limit. That means in 5 minutes from sample A, we have got 10 bubbles. From sample B, we have got 12 bubbles. From sample C, we have got 20 bubbles. But the time limit should be equal for all the samples, right? Because the time cannot be different. So the time should be maintained uniformly. Just to maintain the time uniformly, they have used the stopwatch. Okay. So that you can compare
Yes.
the result in all for all the samples properly. There will be much more accuracy. Okay.
And
Is
ma'am,
it
what
clear?
is the use of this movable lamp?
What is the
Yes.
use of this what?
Lamp, ma'am,
Oh,
movable
that I
lamp.
know.
Movable lamp. Okay, that will provide the light, because without light, how
Yes.
photosynthesis will happen? Light doesn't
Oh,
mean that
Mm.
only sunlight. Okay, there is the
How you will
artificial
take
Yes.
light.
energy?
From the
yes,
light, they will take
Yes.
the photons. Okay, the light contains
Photons.
photons. They will take the
The
photons
chlorophyll
that
will
will help
absorb
them
it.
for photosynthesis. Okay, is
understood,
it
Yes.
ma'am.
clear? So this is why
Why
the
they have used
movable
the
lamp
scale?
is used. Movable lamp means you can, it is movable. That means you can set it in any angle that you want. Okay.
Yes.
So this
Up,
is why the
down.
lamp is used to provide the light for photosynthesis. Now
Why
let's
do
see.
you
So
have
they
used
have
the
taken a
scale?
uniform time of 5 minutes to carry on this experiment.
And why they have used scale? I think.
Explain how the student made sure that the results were due only the change in carbon dioxide concentration. So you will have to tell here that why the student made sure that the results were due only to the change in the carbon dioxide concentration. Okay, so do you have any idea why? You have written this answer, I think,
Yes,
previously.
ma'am.
So why the students are very pretty sure that the change that they can see, the change in the rate of photosynthesis, this is only due to the change in the carbon dioxide concentration.
Because water is given is the means water is giving same amount of water is given to the means used for the all step, and they have separately used the NS means sodium hydrogen carbonate for the means for the 5 steps
Okay,
separately.
so you
Yes,
can
ma'am.
see here that because what is the answer, that because except sodium hydrogen carbonate concentration, all the other means that are used here, all the other components that are used
Same
here,
amount.
they are
We
kept
can also say
uniform.
limiting factors.
They are kept uniform in all the samples. The amount of water, the
Yes.
temperature of water, the lamp, the setup of the movable lamp, everything is kept constant or kept uniform, same
The
in all the
distance of
samples.
lamp and
The
beaker
only
also
thing,
constant.
that varies is the concentration of sodium hydrogen
Sodium
carbonate.
hydrogen carbonate.
The only thing this varies in the 5 samples, and all
That's
the
why.
other components that are used, they are kept uniform. Okay, they are same, their amount, concentration, even the temperature. You can see they are using a thermometer. That means even the temperature they are using, that is also kept constant for all the other samples. This is why they are very sure that the change in the rate of photosynthesis will be due to the change in the carbon dioxide concentration.
Yes.
Okay, is it clear? Is there any more problem?
Yes.
Arijit, you were asking
Yes,
for
No.
this.
ma'am. I have one
Understood.
problem.
And
Page
then what is
number.
the
Let's
use
have
of this ruler, ma'am? Ruler?
Yes.
Let's
For
see,
past and present.
I'm telling you. Let's come to the next part. Carbon dioxide, here you can see what is given. The student repeated the investigation at each concentration and calculated the rate of photosynthesis. Okay, the student repeated the investigation at each concentration and calculated the rate of photosynthesis. The student's results are shown in the table below. So first, in the first column, you can see carbon dioxide concentration. What is the concentration of carbon dioxide? That is controlled by the students. How?
By the by controlling the concentration of
By
Na,
By
HCO3.
controlling the concentration of sodium hydrogen
sodium
carbonate.
hydrogen carbonate.
Now the rate
By
of
giving more, by
photosynthesis,
giving less.
first, second, and third. Here you can see the results that are given. They have repeatedly taken the results. No, here is another one. Another column is also there. Okay, first, second, third, and the mean value they have taken. Rate of photosynthesis. So first one is 3, second one is 2, and third one is 4. What are these number? 1, 2, 4. Sorry, 3, 2, 4.
Rate of the bubbles.
Number of
Yes.
bubbles.
Number of the bubbles per
Per
minute.
second, I think.
Per
Per
minute.
minute. The number of
per
the bubbles per minute. Okay, so they have counted it and then they have taken a mean value of that. So from that mean
In
value,
fast-
why they are taking mean value, why they are counting it 3 times and then taking the mean value? They can count it only 1 time.
And to get more accurate results, and that will help the
Very good. To
students
compare it
other.
and
more accurate
to get the
results.
more accurate results,
And
they
we
are
are telling
counting
the
it 3
average.
times. So
We are
that there
telling the
there
average
should
of
be
this.
no mistake. Okay. Now
Average
come
is
to
the
the ruler
best
part.
case.
Why ruler is used? Ruler is used to measure the distance between the lamp and the apparatus that they have made, so that they can use the same distance for all the samples. For all the 5 samples, the distance, as I have told you, that everything is kept constant, same. See the amount
Yes,
of
I have
water,
understood.
the temperature
Why
of
ruler
water,
is used.
and the distance between the plant and the lamp. Everything is kept
Yes.
same. This is why they have
Constant.
used the ruler.
Constant.
Okay, so there is no
Understood.
relation, there is no important value of the ruler in answering the question. Okay, there is no question from that. I think in section B there might be any question. I haven't seen it yet, but this is very important for making this setup. Okay, the distance between the lamp and the plant should be same in all the samples or in all the setup that they have arranged. Okay, so here it is. You can see the rate of photosynthesis, it increases as the carbon dioxide concentration is increased. Now tell me what more doubt you have from this one.
I have no doubt.
No,
I have
Ajit
also no
Dubey.
doubt.
I have.
Any
I
more
have
doubt is
different
there?
doubt.
Clear.
Clear now? The
Yes,
doubt
ma'am.
that you have told me,
Yeah.
you couldn't understand the setup of this
Yes.
apparatus. Is it clear now?
Yes, ma'am.
Ma'am,
Understood.
can you answer question number E, the last question?
Ma'am,
Last question. Okay, let's see what is written there. The student used tap water as the 0% carbon dioxide concentration. Explain why the student recorded some bubbles being produced. The student used tap water as the 0% carbon dioxide concentration. So where the student has used tap water, then or there, why she has used tap water? She has used tap water in place of what? In place of sodium hydrogen carbonate solution. So as there is tap water, there is no sodium hydrogen carbonate solution. So the concentration of carbon dioxide will be 0, because tap water will not produce any carbon dioxide.
yes.
Right?
That
Yes,
one.
Yes.
Yes.
The marks of this question is 1. You have got 1 marks for that. So the sodium hydrogen carbonate solution was providing the carbon dioxide, and as the student has used tap water in place of sodium hydrogen carbonate solution, so the concentration of the carbon dioxide will be 0%. Okay.
Okay, ma'am. Understood.
Ma'am, I have
And the
problems on
class
page
time
number
is also
30.
over for today. Let me take the attendance first. So in the next class, we will start the